Great. Thank you very much for the introduction. Let's see if I can. Yeah, and I just want to say thank you very much to Simona and Francesca and to the whole organization. This is an outstanding meeting, an amazing venue, and I'm very honored to be here to talk to you today about our research. Often when I give these talks, I start with this slide. There's a sister organization called the Gould Syndrome Foundation in the United States, and they started doing this awareness day on, on April 12th to represent type 4 collagen, alpha 1 and alpha 2, again to raise awareness, and they wear gold as their color. And as I was getting ready for my talk, I found this slide on the website that also shows the family association here wearing gold to represent and raise awareness on April the 12th. So I'm going to start using this slide in my future talks. So what I want to do today is look forward into the future and imagine where are we going to be in 20 years? What will treatment look like? What will a cure look like? Because if we can't imagine where we want to be, it'll be very hard to get there. We need a clear path. And so I want to talk about 3 questions today. How do we get there? So a little bit on what are type 4 collagens and what do they do? Who are we talking about? Who's going to be treated? And then how will they be treated? And some of these things have been talked about already. So in the interest of time, I'll try to go quickly through those sections that I don't want to repeat. But, yeah. So what are collagens? Okay, the collagen superfamily is a family of proteins, and in humans and in mice, in vertebrates, there are 28 kinds of collagens. Okay, and sorry, I'm going to do that a lot. What makes a collagen, as we've heard, is this repeat of glycine XY, gly-XY, gly-XY. And you need glycine at every third amino acid because they fit into this amazing structure, this triple helix, this collagen triple helix. So if you have a gly-XY repeat and make a triple helix, you're a collagen. And despite being called type 4 collagen, it's actually the ancestral collagen. So it goes back about 750 million years ago when single-cell organisms started sticking together and becoming multicellular organisms and making an extracellular matrix. Type 4 collagen was on the scene. And so it's present in every member of the animal kingdom, in all tissues and in all organs. And so what's amazing then is we really don't have a good idea of what their function is, but clearly they must be doing something. And at some point in evolution, collagen 4 alpha 1 duplicated to make collagen 4 alpha 2, and then this whole locus duplicated again twice to make collagen 4 alpha 3, alpha 4, and alpha 5 and alpha 6. And so collagen 4 alpha 1, 1, 2 heterotrimers are ubiquitous. They're present, as I mentioned, throughout the animal kingdom in all basement membranes. Collagen 4 alpha 3, 4, 5, and 5, 5, 6 are much more spatially restricted. And so in 1990, Carl Tryggvason identified the very first mutations in human mutations in a type 4 collagen, so mutations in collagen 4 alpha 5 that cause Alport syndrome, the main consequences being renal disease, because that's where this network is predominantly expressed. But as I mentioned, alpha 1, 1, 2, this network's everywhere, and yet we really don't have a good idea of what it does. So collagens are made in the ER and transported through the Golgi and secreted into the basement membrane where they polymerize and make these sheets, these flexible sheets. But they somehow affect cells, but we don't know if there are 5 functions or 50 functions or 500 functions. We're starting to get an idea of what do things look like if you have a mutated collagen or if you don't have enough collagen, but we really don't have a good idea of how it executes those functions. And so the answer to the first question is quite simple. What are type 4 collagens? The answer to the second half of that question, what do they do, isn't very simple. And we need a lot more research in this area if we're going to develop mechanism-based interventions. So who will be treated? And this is, Elizabeth, I loved your talk today because this is one of the points that I would like to make, is that right now our understanding of what this disease looked like is completely based on glycine missense mutations, but this isn't the end of the story. And so this was work that I had done as a postdoctoral fellow, I was actually interested in studying eye development and ocular dysgenesis and how it leads to glaucoma. And we performed a mutagenesis screen in mice, and we found this strain of mice that had this abnormally developed eye. And based on this phenotype, we narrowed down the chromosomal region and identified mutations in collagen 4-alpha-1. But as we were doing this, also recognized that it wasn't just an eye developmental defect, that these mice also had intracerebral hemorrhages, shown here, perinatal hemorrhages, and also these large cerebral cavities, porencephaly, which we now know. And in fact, this origin story has an Italian twist to it. So and I remember very clearly, it was a Saturday early in 2004, and I was at the lab and I read this paper by Umberto Aguglia and Peter Hutink, and there's a family in southern Italy that had 3-generation inheritance of porencephaly, and this is very rare. You don't usually get multi-generational inheritance. And we'd been studying these collagen mutations in mice for about a year, and so we called Dr. Hutink and we said, I think we know the gene that's mutated in this family. And so sure enough, we sequenced this family, and they had a mutation in one of these conserved glycine residues. And then, the following year, they had a few other porencephaly families. They started identifying more mutations in these conserved glycine residues. And as I mentioned, we knew that the pathology wasn't restricted to the eye or to the brain. So we looked in the retinal vasculature, and there was this abnormal retinal vessel patterning. And we studied the kidney a little bit because of the connection with Alport syndrome that I mentioned. And so if you do transmission electron microscopy, this is in a wild-type mouse. This is the glomerular basement membrane. And we found that the basement membrane was a little bit thinner. There was these focal variations in thickness, and that they had highly penetrant albuminuria. And through a collaboration at the time with Dr. Tornado Serve, they had a family with cerebral small vessel disease that had multifocal recurrent hemorrhages, retinal hemorrhages, and vascular tortuosity, and hematuria. And once again, this family had a mutation in a conserved glycine residue within the triple helix forming domain. And then 1 year later, there was a landmark paper by Dr. Plaisier and Pierre Ronco looking that really sort of put an exclamation point on the fact that this is a multi-system disorder. And I won't go into that further because I know that Dr. Plaisier is going to be talking later today. So again, we now know that there's pathology in many organs, and in fact, in mice, there's pathology in every organ that we've looked at. And so one of the points that I want to leave you with is that there's a lot more to this than what we're currently discussing. The similar slide was just shown earlier. This is sort of the genetic landscape. This is an old slide. I haven't updated it. There's about 50 or 60 mutations on here now. There's many, many more than that, but the landscape is the same. There's a few mutations in this NC1 domain, and then a whole bunch of mutations in conserved glycine residues within the triple helix forming domain. I want to say that for expediency, I usually just say collagen 4-alpha-1, but by and large, everything that's true for collagen 4-alpha-1 is true for collagen 4-alpha-2. However, as has been alluded to, there probably are some biological differences. Part of this discrepancy in the frequency of COL4A1 versus COL4A2 mutations is probably an ascertainment bias. A little bit of that will be going away now that people are doing whole exome sequencing. In the olden days, when you had to just pick a gene or two, collagen 4-alpha-1 was being screened, collagen 4-alpha-2 wasn't. There are even some custom gene panels by GeneDx and Invitae. If you order a cerebral small vessel disease panel, both genes are on. If you order a connective tissue disorder panel, collagen 4-alpha-1 is on there, but collagen 4-alpha-2 is not. So there's probably some ascertainment bias there, but we also think that there are biological differences between these. This part I will go through quickly because it's been covered by many other people. It's simply to talk about the phenotypic spectrum and the number of different organs. This is to point out, again, we study mice because it's a powerful genetic organism in which we can very much simplify some of the complexity that leads to the variable expressivity in humans. And the mice recapitulate the human disease very well. And so who will be treated? As I mentioned, I sort of just went through the phenotypes that we've talked about this morning that are caused by glycine missense mutations. But this certainly isn't the end of the story. So for one point that I mentioned, there are many other phenotypes that we know of in the mice that really aren't represented yet in the literature in humans. And just to give one example, we've been looking at skeletal development. So there's delayed ossification during bone development, and there are thinning of the cortical bone. There's reduced density of trabecular bone. And so again, this is just one example of I think there's many other phenotypes out there that just haven't really gotten the press yet. Secondly, not all of the phenotypes are going to be congenital or developmental. This is some work that we published last year with our collaborator Scott Early. There was a series of 3 papers, and essentially the point here is that we found in these mutant mice that there is age-related cerebrovascular dysfunction. So age-related is the key point here. So these mice have normal cerebrovascular function at 3 months of age, but by 12 months of age they don't. This particular experiment is looking at myogenic tone. So as we increase the pressure inside a blood vessel, the smooth muscle cells should constrict. And in young mice at 3 months old, they do. And in 12-month-old mice, they do not. This is a similar experiment looking at neurovascular coupling. So in this experiment, they're looking at blood flow in the cortex, the whisker barrel cortex. And so when you stimulate the mouse's whisker, you should see increased blood flow to that part of the brain because it's active. And if you look in young mice, this response is intact. And if you look at 12-month-old mice, the mutant mice don't have appropriate neurovascular coupling. So again, these are age-related diseases, not related to any sort of developmental abnormalities. The next part of my talk is essentially what Dr. Turnley-DeServe has pointed out very nicely. So if you do genome-wide association studies for signs of cerebral small vessel diseases, there was 2 very large studies published semi-recently, 2020, both of them, using 50,000 individuals. They found a number of loci. There's 10 to 20 loci that are associated with cerebral small vessel disease. But collagen 4 alpha 2 is strongly associated, and repeatedly associated. And so what this means, genome-wide association studies they're not built to find rare, severe mutations in coding parts of proteins. They're designed to find common variants that sort of predispose to the genetic landscape. And the fact that collagen 4 alpha 2 is coming up in these hits suggests that there's much more to it than these severe glycine missense mutations. And, as Elizabeth pointed out, we think that PADBAL might actually be telling us, it might be giving us a tool with which to understand how changing levels of collagen 4 alpha 1 or collagen 4 alpha 2 might be contributing to these age-related sort of cerebrovascular health and cerebrovascular disease. And so this is the pedigree that she talked about that was published first by this German group, and then it's an enormous 6th-generation pedigree. And then her group published in 2016 that there were these mutations in this miR29 binding site, this conserved binding element that leads to de-repression or increased levels of the type 4 collagen alpha 1 transcript. And so, as Elizabeth also alluded to, we've now made a mouse. At the time, they had identified mutations in 3 of these nucleotides in this conserved binding element. And so we made a single mouse that has mutations in those 3 nucleotides. And this is now a little bit of preliminary unpublished data, but when we do MRI of these animals, we do see signs of cerebrovascular disease. And here's our friend Scott again, who's done some investigations of the myogenic tone. And sure enough, these mutant mice have failed myogenic tone. We actually have 2 independent strains of mice, one that carries the 3-point mutations and another one that has the whole element deleted. And they both show a similar phenotype. And so to summarize this first part of the talk, collagen 4 alpha 1 and alpha 2 are the ancestral members of the collagen superfamily. They're present in all tissue and organs throughout the animal kingdom, but their biological functions aren't known. the mutations cause this highly variable multi-system disorder, but I think we're really at the tip of the iceberg in thinking about, how we're defining it now, and certainly these non-coding mutations. And I'd also like to point out, somebody mentioned earlier about you're sequencing patients and you get VUS, variants of unknown significance. We have a really good idea that mutations in glycine are going to be pathogenic, but we have no idea what happens when you mutate the X or the Y position. And it's almost certain that these things are going to cause problems, but we don't know what collagen does yet, so we don't know what an X or a Y position mutation is doing. So I think there's a whole area there that needs to be explored. So finally, how will they be treated? So just tell you very quickly a little bit about collagen biosynthesis. As I mentioned, collagen 4-alpha-2 will find 2 collagen 4-alpha-1s and make a nice heterotrimer and be secreted. These are dominant mutations, so that means that the patients are heterozygous. So if you assume that they randomly assort in the ER, collagen 4-alpha-2 will find 2 normal collagen 4-alpha-1 proteins a quarter of the time. And then collagen 4-alpha-2 will find 2 mutant COL4A1 proteins a quarter of the time. And half of the time it'll find 1 mutant and 1 normal protein. And there's a little bit of oversimplifying here, but we think for the most part, these heterotrimers don't get out of the cell. And these heterotrimers are normal, so they should all get out of the cell. But we don't know the fate of these heterotrimers. And as also we've talked about, there's this enormous variability. And so what we're trying to do is understand what are the things that underlie the variability? What are the factors that determine where a particular individual will fall upon the spectrum? Because if we can understand those factors, then we can hopefully modify them and take somebody who might fall on the severe end of the spectrum to be more on the mild end of the spectrum. And so there are environmental factors, some of which have been talked about. So for example, these are all in mice. If we performed surgical delivery of pups, they had less cerebral hemorrhage than pups that were naturally born vaginally. If we run the animals acutely, so run them on a treadmill, it increases the severity of intracerebral hemorrhage. And when we put them on anticoagulants, we actually stopped this experiment because within the first few days, 2 of the animals had fatal cerebral hemorrhages. And so these are examples of environmental factors that can contribute where you fall along the spectrum. But there are also genetic factors. And one that I really want to point out, because I don't think it's been mentioned yet, is genetic mosaicism. Okay? We're talking about, often we have an unaffected parent that carries the mutation and has an affected child. I think that we need to really consider the level of genetic mosaicism. Also, if you're trying to compare even 2 individuals with de novo mutations that have the exact same mutation, the timing really matters. So when the mutation occurs will determine how much of their body has mutant cells and how much has normal cells. And this is especially true with extracellular matrix proteins, because we know from experiments where we knock the mutation only into one cell type or only into another cell type, they're actually fairly normal because the tissue doesn't seem to care what the source of the collagen is. And so if you have levels of genetic mosaicism, it can easily determine levels of expressivity. A second cause or contributor to the variable expressivity are genetic modifiers. So this is an example of it. This is a mouse eye, and this is a cross-section of an optic nerve. Both of these are from a mouse with the exact same mutation. They're just on different genetic backgrounds. So this mouse is on a C57 black 6 background, which is very sensitive, and then it's been crossed just a single generation to a different background, and it completely rescues the phenotype. You can't see that slide here, but if you just look at the graph, this is the same experiment looking at intracerebral hemorrhage. So mutant mice on a black 6 background this is the level of intracerebral hemorrhage. If we cross it just a single generation to a different background, it's profoundly rescued. If we cross it to a third genetic background, it's back up here. So there's something in this genetic background that's rescuing the phenotype. And we actually did a genome-wide scan, and we're able to find a single locus on chromosome 1 that confers this phenotypic rescue. And so again, if we can understand what is it about this modifier gene that can protect against intracerebral hemorrhage, maybe we can develop a drug that does this. So that's showing the difference of genetic background. Those were the exact same mutation on different genetic backgrounds. But there's another element. What about allelic heterogeneity? Do all mutations behave the same way or not? And so to answer this question, we have an allelic series of mice. These are all different strains of mice that we have. And we asked how well is the collagen secreted? And I will try to go a little bit more quickly and show you that there is a position effect. So for the mutation here that's in the NC1 domain, it's secreted fairly efficiently. Mutations that are in the triple helix forming domain but near this carboxy terminus, they have more severe intracellular accumulation than do the mutations nearer the amino terminus. So there is this position effect. And then if we take each of those strains of mice, we age them out to 8 months, and then look at the extent of cerebral hemorrhage, and this is what we found. So if you compare, what we see is that there's 3 possible genotype-phenotype correlations. First, this domain effect, where the mutation in the NC1 domain secretes relatively efficiently and has very mild cerebral hemorrhage. This position effect, where mutations nearer the carboxy domain have more intracellular accumulation and more severe intracerebral hemorrhage. There's also an allelic effect that we don't quite understand, where this deletion of exon 41, it respects its position. It has the same amount of intracellular accumulation as its neighbors, but it has a disproportionately severe intracerebral hemorrhage. So there's something there that we definitely need to understand more. But this suggested to us that if this population of heterotrimers is always getting out and this population's never getting out, then the difference between the severe mutations and the mild mutations must be due to this population. And so can we change the fate of this population from those that are being kept inside the cell or those that are being secreted? And so you can do this using a chemical chaperone. So a chemical chaperone is just a small molecule that promotes protein folding and protein secretion. And so when we treat the mutant mice with this chemical chaperone called 4-phenylbutyric acid, this is looking at a retinal vessel of a mutant mouse, and collagen is labeled in red. And so in this animal, you can see that the collagen's all accumulated within these cells on the blood vessel. And if we treat them with 4-PBA, you don't see this intracellular accumulation, and instead you see more uniform labeling of the basement membrane. And sure enough, if we look at the extent of cerebral hemorrhage in the mice that were treated with 4-phenylbutyric acid, it's significantly improved. So again, this tells us that, and I put in air quotes here, the rule, because again, it's much more complicated than what I'm sort of synthesizing here. But we think that in general, sort of the primary mechanism is this impaired secretion, and if we can promote secretion, then this would be beneficial. And the beauty of this is it doesn't matter if there's 5 functions or 50 functions or 500 functions. By promoting secretion of this protein, it'll simultaneously fix all of them. However, this also comes with a caveat. In the same mice that I showed you where we quantified intracerebral hemorrhage at 8 months, we also took skeletal muscle and looked at non-peripheral nuclei as a marker of skeletal myopathy. And this is what we found. It largely follows similar trends, except for this guy, this mutation G394V. This mutation is down here. It's the N-terminal most mutation. It secretes fairly well. It has mild cerebral hemorrhage, but it has the most severe skeletal myopathy. And when we treat those mice with 4-phenylbutyric acid, which should get more of it out of the cell, it actually exacerbated the skeletal myopathy. So these treated animals have more severe phenotype than the untreated mutant animal, and that's shown here. And it seems to be sort of specific to this mutation. So if we take a different mutation and treat them with 4-PBA, it actually helps the skeletal myopathy. And so if you look carefully, you'll see that this mutation, G394V, is sitting right beside this little green bar. This little green bar is an integrin binding domain. And so we think that this is telling us that integrin binding is involved in the skeletal myopathy. And so the broader implication, though, of this is that we need to be very careful about treating people with chemical chaperones. It might work for, I'm going to make up numbers, 80% of people, but 20% of people might have mutations in or near functional subdomains, and we will actually exacerbate one or another phenotype in those people. And the problem is, as I told you, we don't know what the functions are, so we sure don't know where the functional subdomains are. Ideally, in the future, we could map all the functional subdomains, we could genetically stratify the patients and say these ones are safe to treat with chemical chaperones, and these ones aren't. So until that day, we now need to think about what could we do for those individuals? What if we knew more about the downstream targets, and are those areas where we can try and intervene? And so I'll cut this story a little bit short, but we've recently identified that the mutant mice have elevated TGF-beta signaling. So this is probably the best representation of it. This is using a genetic reporter of TGF-beta signaling, and this is a mutant animal and a wild-type littermate. And then if we give these mice 4-PBA, it significantly reduces the TGF-beta signaling. And if we cross them to a mouse that has only one copy of TGF-beta1 instead of two copies of TGF-beta1, so we genetically reduce this pathway, it significantly reduces the amount of intracerebral hemorrhage. And so we think that this is, these are just the papers we published last year. This is also, what I just showed you is also true for the developing eye. And so we think that this is a useful downstream target. So if you have individuals who you don't want to give a chemical chaperone to, you maybe could target TGF-beta signaling. But again, the problem is there might be lots and lots and lots of downstream functions, and it's probably not efficient to try and individually treat all of them. And so if we work backwards, and in a moment I'll turn the floor over to my colleague, Patty Mussolino, but gene editing is probably where we really are going to be in the future. However, there's always a caveat. Gene editing and also chemical chaperones can only work if the collagen is still being produced. So another wrinkle about collagens is they're highly, highly stable proteins. Type 1 collagen, the half-life is estimated to be over 100 years. Type 4 collagens, we don't really know, months to years. And so if these things are very, very stable, then chemical chaperones or gene editing might not be very effective because the collagen's not being turned over. And so timing is really going to be of the essence. And so how will they be treated? I think I'm trying to show you that there are multiple steps in the pathway, and we're still trying to understand each of these mechanisms that could represent therapeutic targets. I will not go over my summary slide because I just spent all this time telling you. I do want to thank the members of my lab, our collaborators, our funding sources, and these are the folks in my lab. And once again, thank you very much for organizing this fantastic meeting. I'm happy to take questions.